move on to the third part of this module. So here we are, genetic technologies. Does artificial manipulation of DNA have the potential to change populations forever? So here we go. We're going to discuss the uses and advantages of current genetic technologies. We're going to talk about reproductive technologies, cloning, recombinant DNA technologies, benefits to agricultural and medical and industrial applications, effect on biodiversity of agricultural biotech, influence of social, economic, and cultural contexts, and our inquiry question is, does artificial manipulation of DNA have the potential to change populations forever? So here we go, genetic technologies. This is what we are going to look at. Now, the key here is that some of them are listed specifically in the syllabus, but I'm going to talk you through them, and I just want you to know what it is, how it works, know examples for it. And I think even if you want to like draw down this little table, I think it's a really good visual of seeing actually what each one is. What is the reproductive technology, what is cloning techniques, and what are the recombinant DNA technologies that we're looking at. So let's begin by looking at artificial insemination and pollination. We've already talked about this in module 5. So artificial insemination is the injection of semen into the uterus without sexual intercourse. So we select animals that have favorable traits, and we basically take the egg and sperm, fertilize it, and we can also, what we can do is we can fertilize birth cycles. We can synchronize birth cycles in order to essentially synchronize across however many number of organisms that are being produced, and it can also lead to reduced injuries. We then have in vitro fertilization. So in vitro fertilization, IVF, is when the egg is fertilized outside of the body. So this is a fertility treatment that is used by humans to achieve successful pregnancy, and it also allows for embryo screening to occur. So here, for example, the process is that a woman is given hormone treatments to stimulate the production of eggs. So essentially, what happens is, once that happens, multiple eggs are taken from the woman's ovaries right there. In the labs, the eggs are mixed with a man's sperm and they're cultured to become fertilized. Once they are fertilized, they're placed in an incubator for 48 hours. And then they're planted in the woman's uterus, or they're frozen for future implanting. Okay, here we go. So reproductive technologies, artificial pollination. So plant sperm is purposefully taken from another plant, and it's placed in the stigma of another plant. So if you want to know these terms again, it is it is a really good revision for Mod 5 as well. So it leads to the creation of new plants with favorable traits, so pure breeding, or we can also have crossbreeding, and genetic experiments, of course, like Mendel's experiment can also occur here. Now, putting it all together, here are the outcomes. Artificial insemination leads to the passing on of favorable traits and favorable genes to the offspring. Increased efficacy of livestock industry, however, it can lead to limited genetic variability. In vitro fertilization, able to reproduce with desired mate, it allows for genetic screening of embryos to avoid disease, however, it is expensive. And artificial pollination leads to higher crop yields, selection of desirable traits, and also to the creation of monocultures. Okay, another practice question. I've got the responses there for you. Have a look, have a go at these questions. It's a really good practice. Okay, let's now look at cloning. We're going to talk about whole organism and gene cloning. So, whole organism cloning, biggest example is the very famous Dolly the sheep. So it uses somatic cell nuclear transfer. So somatic cell nuclear transfer involves the removal of an adult cell from the organism that we want to clone. We then take an unfertilized egg from a donor and we denucleate it, so it's basically like an empty vessel. The denucleated egg and the stem cell are fused together, then the cell is cultured, it begins to divide. We then plant that embryo into a surrogate organism, and then the surrogate will give birth to an organism. Effectiveness is that offspring may not be strictly identical to the parent. There can be somatics or mutations that can occur in the offspring. There can be mitochondrial mutations as well, remember, because the mitochondrial has its own genetic information, and epigenetics is also part of it. It is an expensive process, and it's not time efficient, and it is better to just clone the organs of an animal rather than to clone the whole organism itself. Gene cloning. So gene cloning, how do we do it? So we essentially are creating lots of copies of a gene of interest, and where is my, there we go, highlighter, lots of copies of a specific gene. Okay, so essentially we assemble recombinant DNA, so we identify what the target gene is, we cut the gene out or order it online, so we cut the gene out, we then treat that gene fragment and plasmid with restriction enzymes. So we treat it with restriction enzymes here, and what that means is we cut the gene in specific places, and then we create sticky ends of that gene. We then combine the sticky ends together, and once the sticky ends come together, they come together by base pairing affinity, and then we treat with DNA ligase to repair the backbone. The replicate, so that's the first bit there. We remember, DNA ligase should sound familiar from DNA replication, where the ligase joins together the Okazaki fragments. DNA replication, so after we've done that, we insert the recombinant plasmid into a host bacteria. Bacteria is used because they are super microfactories, like they can replicate really fast. So we essentially put that plasmid into the bacteria. It will express the gene and produce lots of proteins. And again, if you want to actually get a large number of the DNA copy, then we don't, and not the protein, because the DNA will produce the protein. But if you want to get the DNA copies itself, we can use polymerase chain reaction, which is basically going to spur like thousands of copies of the DNA. So here is the process. There's the gene, there's the plasmid. So essentially there's the plasmid, there's the DNA, we've got the bacteria, we've got a plasmid, which is a circular DNA plasmid. Here's the bacterial plasmid. We're going to put a restriction enzyme in it so that we can cut it open at a specific spot. We've got the linearized DNA with our target gene, and we're going to then put the two together right there, and that's going to lead to a recombinant plasmid. We're going to pop this into bacteria, and this has been used to produce insulin. So in the treatment of type 1 autoimmune disease, type 1 diabetes, where the person's body does not produce insulin, which means they can't regulate their blood glucose levels, and so that's where we can use this process to produce insulin and help them, yeah, essentially regulate their blood glucose levels, and as a result survive. So effectiveness is that it's fast, it's cheap, bacteria is easy to work with, and it is also easy to buy and sequence DNA. Okay, recombinant DNA technologies. Let's have a look at how we, this is essentially recombinant DNA technologies. It's a method to join together DNA from two different species in order to produce new genetic combinations. So transgenesis is the process through which we introduce exogenous genetic material, which is like material from outside the person, into another organism. And it is performed so that the organism can exhibit a new trait. And remember, DNA is ubiquitous, it's the same across organisms, so we can insert new genes into genomes. And the way we can do it is we can either do it through plasmids, retroviral vectors, or DNA microinjections, which is like a fine glass needle. So this can actually be done for agricultural and medical applications. So what do? We begin by identifying a gene of interest. We extract that gene right there. We take it and we're going to deliver it, and we're going to put it in a culture, and then we are going to lead to, and then we can grow it so that we can have a genetically modified crop right there. So how do we make a transgenic organism? The first thing we do is we identify our genes, then we get large amounts of a gene of interest, and then we insert the gene into a target organism. The first technique to get to identify our genes is gene sequencing. This is what we looked at DNA sequencing in module 5, so I'll just skip through this, but it allows us to gather single nucleotide information. We can identify genes with useful traits, and we can use that. The second step is basically using PCR. Either PCR or gel, either PCR and then gel electrophoresis. So PCR to amplify the DNA, to get lots of copy of the DNA, and then we're going to use gel electrophoresis to visualize the DNA to check that we have the right type. So we're going to get large amounts of it, but we're going to check that we have the right type, and then we're going to insert it into a target organism. We can do it through restriction enzyme cloning, so what we looked through in that earlier example, where we basically get the target gene, we get the plasmid, and we basically put it together and we get a recombinant plasmid, and we're going to put it into a bacteria. We can also use transfection, which is basically we're using a virus as a vector. We're going to use a virus as a vector to put the gene into the target cell. Remember, viruses, they don't have their own machinery to produce proteins. They will go into what is going to happen is we're going to use the virus as a vector. So we're going to put a gene into the virus. We're going to send the virus into the target cell, and the virus is going to hijack the machinery of the target cell. It's going to then lead to the target cell. It's going to ask the target cell, basically force it to then make the proteins of that specific gene that we wanted to make, because that's what viruses do. But this time, instead of having the viral proteins in there, we're going to have the genes that we want. And finally, microinjection. We use a very fine needle, and we just, because the cells are small, and we just inject the DNA. Okay, here we go. That's the whole process. I will leave that for you to have a look at. Let's talk about some examples. So agricultural example, BT cotton. BT cotton, so basically the Bacillus thuringiensis produces Bt toxins, which are insecticidal, so they kill moths, beetles. And what we have done is the Bt toxic gene is inserted into cotton crops, because when pests eat the crop, the toxin is released and the pest is killed, and that reduces the need for farmers to actually use insecticides. The medical example of E. coli that we just talked about when we talked about recombinant DNA. So we know that insulin is an essential hormone which aids in the regulation of glucose metabolism, and inadequate production of glucose can lead to the development of diabetes. And what we do is we insert human insulin into E. coli, which is a bacteria, and we allow it to produce large amounts of it. We then take it, we purify that protein from the bacterial cells, and we use it to treat diabetes. Here we are. Recombinant DNA technology. So gene therapy is the correction of disorders. So we can actually correct disorders by introducing changes into the functional genes, and we can introduce healthy genetic material. The cell and its offspring can then have restored function, and we can insert using viruses, gene guns, or inorganic nanoparticles, and it can happen either in vivo in the living thing or ex vivo. CRISPR-Cas9 is a technology that actually edits the system for accurately introducing point mutations. So we introduce point mutations to fix something in the genome. So it has two parts. It has the guide RNA, which contains the nucleotide sequence, and it has the Cas9. The guide RNA is basically going to contain the complementary sequence that we want to edit, and the Cas9 is an endonuclease. It's actually going to cut the DNA so that the guide RNA can get in there and fix it up. ELISA is enzyme-linked immunosorbent assay. So what that is that it's a biochemistry tool which detects the presence of antigens in a liquid sample. So what it does is it's a diagnostic tool which exploits natural antigens antibody interactions. So within our bodies, we have this antigen antibody interaction, and what happens is that the levels of biomolecules in a sample can be measured by linking levels of antigens or antibodies to a fluorescent readout, and it's useful for forensic epidemiology, and we can trace the spread of infectious diseases using this. So all our cells have antigens, and those are like our body antigens, like our self antigens, but every time we encounter a virus or a bacteria, they have their own antigen, and the body recognizes that as like an outside thing, right? Like it's a non-self particle, and then the body has its own antibodies that it uses to latch onto the antigens and identify those antigens as non-self. But these are the three right there. I would recommend watching like actual videos of how these technologies work. You can find some really good ones, because that will give you like a visual of how they work. I know we're going a bit over time here, so I'm going to finish up. I'll leave those practice questions for you. Last bit, here we are. So agricultural techniques and benefits. So techniques include selective breeding, artificial pollination, transgenesis. Benefits are that it leads to the creation of crops and livestock with favorable traits, high yields, high nutritional value, better temperament, and also reduced environmental impact. They can create organisms which do not require insecticides or herbicides, and it increases food security. In terms of medicine, there can be cloning, gene sequencing, gene therapy, CRISPR that we've looked through, and ELISA. Benefits are we can have personalized medicines. They can be preemptive diagnosis, treatment of genetic diseases, potential cures using like CRISPR-Cas9, and gene sequencing, improved diagnostic tools, cheaper, faster treatments, and creation of molecules which can be used for medical treatments like insulation for diabetes. Industry techniques, gene cloning, sequencing, transgenesis. So benefits are we can have increased speed of chemical reactions, more efficient industrial processes, creation of many everyday items, and creation of organisms which produce industrially significant products like biofuels, biomaterials, and energy. I'll leave the positives and negatives for you. They're very similar to what we have discussed. I will also leave the social, economic, and cultural context for you, because this is just putting together what is the influence of social, economic, and cultural context on biology, on these biotechnologies. So that includes, like looking at the big picture of countries, law, environment, religions, economies, and bringing that down to the science and the students and the relationship between the two. So that's there. These examples I will leave for you to have a look at, and this is what we have covered, and these are some resources for you to have a look at if you want to get some more examples for genetic technologies.